Hi everyone, in this video I'm going to do a debrief on operons. Operons are clusters of genes. They're found only in prokaryotes because prokaryotes have the ability to do transcription and translation at the same time since they don't have a nucleus. Eukaryotes don't have that luxury because with eukaryotes the DNA is found in the nucleus and transcription must be completed, including all the processing of the primary transcript before the mRNA is allowed to leave the nucleus into the cytoplasm where translation will take place. But prokaryotes don't have that issue because they don't have a nucleus, so transcription and translation occur at the exact same time. So an operon, in its most generic form, is a cluster of genes along a segment of DNA. So let's say that this horizontal blue line represents a segment of the prokaryotic DNA. Now, within this segment of the prokaryotic DNA, you would have several genes, which I'm just going to label very generically as A, B, C. And these genes are each going to make a polypeptide that come together to form a functional protein. But in order to make the protein, the genes have to be transcribed and translated. Now, an operon is not only going to include the 3 genes that make up the protein, but there's a section before the operon called an operator. And in front of the operator is a segment that is called the promoter. So you could think of the promoter like the origin of replication. It's similar in that this is where the RNA polymerase is going to first bind onto the DNA to begin transcribing the base pairs. So the RNA polymerase is going to find the promoter, attach onto it, move forward, and at this point right here is going to begin making the mRNA that will eventually make the polypeptide that's going to form this entire protein. So the RNA polymerase is going to move this way to transcribe these genes. Now, the operator is a separate section of the DNA in between the promoter and the genes themselves. And what happens at the operator is sometimes another molecule called a repressor will bind to that section. And when the RNA polymerase tries to get past the operator, if the repressor is bound to the DNA right here, it acts as a stop sign and prevents the RNA polymerase from reaching the genes to make the protein. So this repressor acts as a stop signal because it's bound to the operator here and prevents the RNA polymerase from reading the DNA. So this is the basic overall structure of an operon. Let's talk about the 2 operons y'all learned about in your homework videos. The first type is known as a repressible operon, and the example that y'all learned was the trip operon. So in the case of a repressible operon, the default setting for this gene, or the default setting for the operon, is in the on position. That means that this operon is going to be making the mRNA that's going to eventually code for something. And in this case, it's coding for an amino acid called tryptophan. Well, technically, it's coding for the enzymes that are going to make the tryptophan amino acid. So the default setting on a repressible operon is in the on position. But we can turn this off if the conditions are right. So let's talk about how to turn off the repressible operon. And it has to do with the repressor molecule. So for the repressor molecule, we can also think of the repressor molecule as being on or off. And keep in mind that the repressor is going to turn off this gene by blocking the RNA polymerase. So if this operon is repressible and is always on the on position, make a prediction about what you think the default setting for the repressor will be. Did you make a guess? Well, if you guessed the off position, then you are correct. Now, the reason why it's in the off position is because this repressor will block the RNA polymerase when the repressor is bound to the operator. So you don't want the repressor bound to the operator if the operon is going to be functional. In other words, it's going to be making the mRNA. So for the repressor, the default setting is off, meaning that it's not going to bind to the operator, but it is going to be turned on and then be allowed to bind to the operator if the conditions are right. So in this case, it's tryptophan in the environment. So if tryptophan is available in the environment, it's going to bind to the repressor and turn it on. By turning on the repressor will now bind to the operator and prevent RNA polymerase from being able to transcribe the genes. And it makes sense because if there's trip in the environment, if there's tryptophan in the environment, then there's no need to make more tryptophan. That's just going to be wasteful and using up resources and energy. The second type of operon is known as an inducible operon. And in your homework, you all learned about the lac operon. So the inducible operon is the exact opposite of the repressible. In other words, the default setting for the inducible operon is always off. But these genes can be turned on if the conditions are right. So in this case, what's going to happen to the repressor is the default setting will always be in the on position because it's going to block RNA polymerase from making the proteins or from making the mRNA that will become the proteins for breaking down the lactose. Now, how to stop the repressor or switch it to the off position is by having the lactose present in the environment. I hope this video was helpful. Thanks for watching. Bye.